Hello everyone. Let us study about the respiratory chain inhibitors and uncouplers of oxidative phosphorylation. This is Dr. P. Miriam Kumar, Head of the Department of Biochemistry, here explaining this topic. Outcomes of the lesson would be like by the end of the lesson, the learners will be able to understand about the inhibitors of electron transport chain. Then they'll be able to narrate the mode of action of each of the type of inhibitor. Then explain about physiological uncouplers. Then describe the mode of action of uncouplers of oxidative phosphorylation. The contents that will be dealt as a part of this topic are: first thing, you'll be learning about the inhibitors of electron transport chain, followed by that the uncouplers of oxidative phosphorylation, and we'll be dealing with two types of uncouplers. First, let us study about the inhibitors of electron transport chain. So there are many inhibitors which operate at specific sites along the electron transport chain. They bind to one or the other component of the electron transport chain and they block the transport of electrons. So the presence of these inhibitors, it leads to accumulation of what is known as the reduced components or the reducing components. So as this is happening, you see that ATP synthesis is not taking place. So we have a series of inhibitors that are operating along the electron transport chain. And the key steps where or the key points where these inhibitors operate are mentioned here. You have the NADH and coenzyme Q. At this point, the inhibitors can operate, or inhibition of electron transfer from cytochrome B and C1, or it is even the inhibitors that operate at cytochrome oxidase, and then even cyanide poisoning is one of the reasons wherein electron transport chain is inhibited. First, let us consider the inhibitors which block the transfer of electrons from NADH to coenzyme Q. So the important ones are amylol, retinone and pyridine. These are the ones that inhibit this transfer, thereby blocking the next series of steps to happen. Next one is inhibition between cytochrome B and cytochrome C1. So here you see that you have antimycin A and BAL. Antimycin A and this BAL, it is the antidote actually, which is used in as an antidote against war gas. So BAL, it means British anti-leukocyte, which is an inhibitor for this particular site. Next is inhibition of cytochrome oxidase here, which is done by cyanide, carbon monoxide and sodium azide. So you see that carbon monoxide, it reacts with the reduced form of cytochrome, and while cyanide and azide, these two, they react with the oxidized form, thereby inhibiting the electron transport chain. Fourth one is cyanide poisoning. Cyanide is considered to be a very potent inhibitor of electron transport chain, and it binds to the Fe3 plus iron 3 plus of cytochrome oxidase. Thereby, it will be blocking the mitochondrial respiration and it can lead to death of the cell. Cyanide poisoning in human beings, it can cause asphyxia, wherein most of the central nervous system it will be affected, which is because of the death of the cells or death of the tissues because of cyanide poisoning. So this is the summary of inhibitors. So we have three sites for inhibitors of the electron transport chain. First site is here between NADH and coenzyme Q. Next between cytochrome B and C1. And next the last one, cytochrome oxidase. So here you have amylol, retinone and then pyridine. While at site 2 you have antimycin A and BAL. While lastly you have cyanide, carbon monoxide and sodium azide inhibiting cytochrome oxidase, the last step. So all these are the inhibitors of electron transport chain. Now let us look into the inhibitors of oxidative phosphorylation. We have two types of these things. One we call them as the uncouplers, while the other one we call them as the physiological uncouplers. First, let us see about uncouplers. Uncouplers are the compounds. They'll be uncoupling or delinking electron transport chain and oxidative phosphorylation. That means oxidative phosphorylation is delinked from the electron transport chain. So what happens during this process is uh, there is an increased permeability of the intermitochondrial membrane to protons, and this also disturbs the ATP synthesis process. These uncouplers, they also increase the rate of oxidation of substrate as a result, and this will be happening without ATP synthesis. Okay, without ATP synthesis, which means that there is an increase in NADH and FADH2 oxidation. Okay, this is seen during the presence of uncouplers, but there is no ATP synthesis which is taking place when you add uncouplers. Examples include, the first one is very much known to us, 2,4-dinitrophenol, or which we abbreviate it as DNP. DNP, it is a very small lipophilic molecule and it acts as an uncoupler as it carries protons and it makes them easy to diffuse through the inner mitochondrial membrane. So as such, what should happen is the inner mitochondrial membrane should be impermeable to the protons, thereby contributing to ATP synthesis as the protons are being pumped from the inner mitochondrial membrane to the matrix via FOF1 ATP synthesis, right? But here, because the membrane is getting permeable, the protons are just diffusing through the inner mitochondrial membrane. So here, this particular thing actually, they use it when a person is willing to go for weight loss programs. So one side effect is that it causes lots of hyperthermia, and the FDA has recently banned usage of DNP for the weight loss, especially for weight loss. So it's considered to be little dangerous. Next one is you have other compounds also which are used, like we have this dinocresol, pentachlorophenol, trichloro carbonyl cyanide, and phenyl hydrazone, etc., which are also being used as a part of which are also being used as uncouplers. So here this one they are considered to be very highly potent. They are 100 times more effective than the routine uncoupler, that is the DNP. It is even said that even aspirin, when it is given in high doses, that also acts as an uncoupler. So it will lead to lots of oxidation taking place, but it will not allow ATP synthesis taking place. So that is one more important thing. So these are the uncouplers which are routinely studied. Next we have the physiological uncouplers. So physiological uncouplers means these are some of the substances which are produced during the normal physiological reactions. And these act as uncouplers when they are present in high concentrations. Say for example, thermogenin, we have thyroxine, and some long chain fatty acids. They also act as uncouplers. Let us talk about thermogenin. Actually, thermogenin, it's also known as uncoupling protein, UCP, and it is usually found naturally in the brown adipose tissues of animals which are hairless or which are hibernating or which have adapted to cold climates. So in those animals it is said that it is there, and these are specialized in carrying out uncoupling of oxidative phosphorylation from electron transport chain, and it blocks the liberation of ATP, thereby generating lots of heat. So this is how these uncouplers, physiological uncouplers, will be acting. The other physiological uncouplers are ionophores and vancomycin and nigersin. Ionophores, these are considered to be lipophilic substances, and these promote transport of ions across the biological membranes, thereby affecting ATP synthesis. While we have these antibiotics, vancomycin and then nigersin. These also act as uncouplers. What they do is they promote the formation of proton gradient across the inner mitochondrial membrane, but they inhibit oxidative phosphorylation. It is said that they inhibit the oxidative phosphorylation. Thereby, they will be acting as uncouplers. The references that are used while making this lesson are, I referred to principles of biochemistry, Leninger's principles of biochemistry, then biochemistry by U. Satyanarayana, and Harper's illustrated biochemistry. In this topic, what we have studied is the inhibitors of electron transport chain, the three sites where electron transport chain is inhibited, like the NADH and coenzyme Q, then between the cytochrome B and C1, then at the cytochrome oxidase. Then followed by that, we have studied about the uncouplers of oxidative phosphorylation. So we have some uncouplers as well as physiological uncouplers that we have seen. DNP is the uncoupler which all of us know. Then under the physiological uncouplers, these are found in hibernating animals or animals which live in cold places. The best example is thermogenin. And then we also have studied about ionophores and antibiotics like vancomycin and then nigersin. So thank you for watching the video. Please read the notes and work with the assessment. Thank you.